head.
1, 2, 3.
We need to monitor the uterus. Please open up the abdominal cavity. Blade. Suction. Dr. Chris, there's the uterus. Good job, doctor. Dr. Chris, here's the bicoordinate uterus. Do we need baselines on that? Yes, doctor. Let's get some baseline contractions going. Where's the technician at? Hey, can we get some isometric transducer set up, please, for some baseline contractions?
Right away, doctor.
Go ahead and administer the drugs, doctor.
Doctor, baseline contractions are beginning to show. As you can see, doctor, both the amplitude and frequency appear to be low. There seems to be some issue of some sort.
You know what? You are right. I think we have to increase the contractions as soon as possible. Alright guys, we need to increase the contractions. What do you guys propose?
You know, I remember a drug, it's really popular, Cytotec.
Actually, the rat came in with a bracelet that says that she's allergic to that.
So,
Interesting.
is that not going to work then?
No, it's not. We need a different solution.
What about high calcium solution? I remember back in med school, Professor Chris was talking about how administering high calcium solution can be used to remedy low uterine contractions for smooth muscle.
That might actually work. I remember that too.
Alright, to start things off, are we talking about smooth muscle contraction? Smooth muscle tissue is characterized by the smooth muscle cells that comprise the tissue itself, as well as the autonomic nervous system and the neurons that innervate it. Varicosities in the autonomic neurons contain lots of neurotransmitters, which, under stimulus, release these neurotransmitters in large quantities onto the surface of the smooth muscle. These neurotransmitters bind to surface receptors on the muscle tissue, inducing depolarization of these cells. Numerous cells within the muscle tissue are electrically connected by gap junctions, however, so depolarizing stimulus applied to a single cell is able to rapidly propagate almost instantaneously to all other cells in the same muscle unit, allowing for a unified contraction of nearly all myofibers at once. This is why this kind of tissue is called single unit smooth muscle, because it contracts as a single unit. As you can see, calcium has a variety of effects on contractions. So if I remember back to med school, typical muscle contractions in smooth muscle are usually characterized by M3 acetylcholine receptors in the membrane, which are GQ, GPCR cascade mechanisms. So acetylcholine binds to the receptor and activates this GQG protein. So once it's activated, it turns into and activates the membrane-bound protein called phospholipase C, which converts PIP2 into DAG, or diacylglycerol, in the membrane space, with IP3 produced as a byproduct of the reaction. So a couple things happen from here. DAG moves into the cytoplasm to activate protein kinase C, which has the twofold effect of activating LTCCs and inhibiting potassium channels in the cell membrane. So at the same time, IP3 that's produced earlier gates the IP3 receptors in the membrane of the sarcoplasmic reticulum. So at this point, there are two sources of calcium that come into the cytoplasm that mediate the smooth muscle contraction cascade. So some of the calcium flows through the L-type calcium channels, and calcium also enters from the cytoplasm from the sarcoplasmic reticulum. Calcium also activates the ryanodine receptors to further encourage calcium release into the cell. So some of this calcium also inhibits the LTCCs in order to truncate the flux of calcium ions into the cell. Calcium is then able to bind with a protein called calmodulin to form this complex that activates myosin light chain kinase. So myosin light chain kinase and myosin light chain phosphatase act in opposing fashion to either phosphorylate or dephosphorylate the myosin head groups, with the calcium calmodulin complex primarily activating MLCK. The prevailing result that we see is muscle contraction. If we subject the uterine muscle to high calcium solution, the same exact pathway is activated, just that high calcium in the extracellular space leads to high calcium levels inside the cell during contraction, and that's what allows greater contractility. So Dr. Chris, that's why I think if we administer this drug, we should probably expect to see greater contractility, higher contraction frequency, and longer contraction duration too. The actin and myosin architecture in smooth muscle cells is slightly different from that normally seen in skeletal muscle. The cytoskeleton of these cells is largely dictated by dense bodies, which serve as anchoring points for actin filaments. These dense bodies are connected amongst themselves by intermediate filaments that hold the structure together. Actin filaments are bound to these dense bodies and surround myosin filaments. The myosin filaments themselves are covered in myosin head groups, so in this way they're able to create sufficient overlap with nearby actin filaments at varying levels of muscle stretching. In the actual contraction cascade, the head groups bind to the actin filaments and in a uniform ratcheting motion slide along the filament to create movement, in this case in opposite directions. This movement in large scale across all actin-myosin interactions within a single muscle cell give rise to the contractile activity we observe in uterine smooth muscle. There is a repetitive cycle of interaction between myosin head groups and the respective actin filaments that they undergo to produce the power stroke of muscle contraction. First, the head groups with ADP and phosphate bind to the actin filament. There is a release of these ADP and phosphate groups, during which time the myosin head group executes its power stroke, pulling the myosin filament along the length of the actin filament. Finally, the myosin head group eventually dissociates from the actin filament and after phosphorylation is able to reset to its ready position to be hydrolyzed for the next iteration of the contraction cycle. So what I remember from med school about generic muscle relaxation is that any freely available calcium in the cytoplasm is pumped into the SR through the Circa pump's active transport. So the continually decreasing intracellular calcium concentration encourages dissociation of the calcium calmodulin complexes, which further feeds the process of calcium ion sequestration into the sarcoplasmic reticulum. The consequent imbalance between MLCK and MLCP activity causes MLCP dephosphorylation activity to prevail, which induces smooth muscle relaxation. Calcium ions in the cell inhibit L-type calcium channels and also activate potassium channels, which flux potassium into the extracellular space to restore membrane potential. Another membrane protein called the sodium calcium exchanger, or NCX, antiports 3 sodium ions into the cell against a single calcium ion out of the cell. This is supported by function of the sodium-potassium pump that helps maintain membrane potential and ion balance inside the cell. Finally, calcium ATPases help to move calcium ions back out of the cell in order to conclude the relaxation portion of the smooth muscle contraction cycle.
Go ahead and administer the drugs, doctor.
Doctor, both the amplitude and frequency have increased after the addition of calcium, but they appear to be too high.
There's no way these values can be that high. How did you measure them?
Doctor, we measured it by using the baseline right here, the amplitude right here, the duration right here, and the period used to find the frequency.
Guys, high calcium just induced contractions are way too high. We need to get these down as soon as possible.
You know what? I remember ergotamine actually being used to treat that.
Ergotamine? That's ridiculous! From the literature that I just read, it binds to the 5-HT2 serotonergic receptor, and that increases contractions. How would it even be useful here?
You know, I've seen dubious studies produced all the time for profiteering. But you know what? I personally think that, serotonergic, from the literature I read, that the ergotamine will actually bind to both alpha and beta receptors non-discriminatively. And it acts more as an antagonist than an agonist. It can act as a partial agonist, but I pretty much think that this will decrease the contractility of the uterine muscles. We might as well give it a try.
You know what? I think you both are right. You guys have new research. We don't have time to waste. Let's give it a try. Alright, remember that ergotamine is very useful in smooth muscle physiology, but its mechanism is very complicated.
I remember back in physiology medical lab, we had talked about the rat uterine physiology. Specifically, remember it has a couple of major receptors, the adrenergic alpha-1, alpha-2, the beta-2 receptors. It also technically has beta-1 receptors. But of those, the most prevalent are the beta-2, followed by the alpha-1. Alpha-2 and beta-1 are usually only occur with increasing estradiol levels and with pregnancy, along with like different cycles, such as the menstrual cycle. Since ergotamine is a non-discriminative binding agent, agonist and antagonist to all of the receptors mentioned, it would make sense that the effects would be relatively unknown and would be dependent on the smooth muscle that is being tested upon. However, the primary physiology does not change in any form or fashion. Upon myogenic stimulation, calcium enters into the cytosol from the extracellular space through the LTCC channels, and additionally, this calcium will act upon RYR receptors on the sarcoplasmic reticulum. The increase in IP3 concentration will also bind to the IP3 receptors on the sarcoplasmic reticulum, which will cause a net efflux of calcium out of the sarcoplasmic reticulum, combining with the calcium that has entered through the LTCCs to form your net cytosolic calcium. This net cytosolic calcium will bind to calmodulin, as mentioned previously, and then the calmodulin will react with myosin light chain kinase to phosphorylate myosin. Relaxation would occur via the LTCC channels being inhibited by the cytosolic calcium and the potassium channels being excited by the cytosolic calcium. Then myosin light chain phosphatase would cleave off the ATP phosphorylation of the myosin, which would then break the cross bridges and reduce active tension. And then what would end up happening is the calcium that is free floating in solution would unbind from calmodulin and would be put back into the sarcoplasmic reticulum via the sarcopump and be exported out of the cell via the sodium calcium exchanger proteins, as mentioned previously. The duration of the contraction caused by ergotamine's administration is incredibly variable due to the fact that ergotamine is binding to many different receptors, and as such the effect is not predictable in any form or fashion and is solely dependent on the tissue that is being experimented upon. So the beta-2 adrenergic receptors, which are GS protein coupled, lead to smooth muscle relaxation. This process begins with the activation of adenylyl cyclase and then the consequent conversion of ATP into cyclic AMP. This cyclic AMP will upregulate the activity of protein kinase A in the cytoplasm, which will primarily serve to activate myosin light chain phosphatase, increasing its activity, and inhibiting myosin light chain kinase, decreasing its activity. Protein kinase A also excites the potassium channels, increasing their activity. So the beta-2 adrenergic receptors, which are GS protein coupled, lead to smooth muscle relaxation. This process begins with the activation of adenylyl cyclase and then the consequent conversion of ATP into cyclic AMP. This cyclic AMP will upregulate the activity of protein kinase A in the cytoplasm, which will primarily serve to activate myosin light chain phosphatase, increasing its activity, and inhibiting myosin light chain kinase, decreasing its activity. Protein kinase A also excites the potassium channels, increasing their activity. The GI pathway, which is prevalent on the alpha-2 receptors, works in opposite to that of the GS pathway by inhibiting adenylyl cyclase.
Go ahead and administer the drugs, doctor.
The addition of ergotamine has only caused a slight decrease in the frequency and amplitude of contractions. What do you suggest we do, doctor?
You're right. I'll go find something else that can decrease contractions a little more. As you can see, you both were incorrect, but correct at the same time, whatever that means. But this time is running out, and we need a solution as soon as possible.
You know what I heard about terbutaline, I think is? You know, it's used to stop contractions.
I don't know about that. I think I read recently that the metabolism is really taxing to the liver. We should find a safer alternative for the rat.
I agree with
Safer
Jay.
alternative.
Yeah, I mean, what about dantrolene, for example?
Magical sodium?
It's not glorified sodium. Like, hear me out. So dantrolene blocks the ryanodine receptors in the sarcoplasmic reticulum and should decrease calcium in the cytoplasm. I think it would work in terms of decreasing contractions.
I mean, your logic is fair, but how do you know that to be true?
I've read a lot of papers, and that seems to be what the conclusions are. Do you?
Well, aren't you the academic?
Well, don't you think it's worth a shot?
Maybe.
I agree with Jay. I think he's done his research here. We'll go with dantrolene. Alright, our next topic is the drug dantrolene. Can anyone tell you what it does? You over there.
Who cares? It's just glorified sodium.
Not quite. Actually, dantrolene has a huge impact on smooth muscle physiology.
So from what I remember from med school, dantrolene in the cell migrates specifically to the SR, where it blocks the ryanodine receptors that normally allow calcium to flow out into the cell. Calcium usually moves through the RIR and the IP3, but since the RIR receptors are blocked, there's residual calcium left in the SR, and so there's lower than normal calcium levels in the cytoplasm. Calcium otherwise enters into the cell from the extracellular space through the LTCCs as normal, but either way, there's still less calcium available. Dantrolene binding to the ryanodine receptors is reversible though, so the substrate can unbind from the receptor. Anyway, calcium binds to calmodulin to form the calcium-calmodulin complex, which in turn activates MLCK. Overall, the relatively weaker activation of MLCK gives rise to lower contractility, which is directly derived from lower calcium concentration inside the cell. That's why I think if we administer this drug, we should be able to expect to see a decrease in uterine smooth muscle contraction activity.
Go ahead and administer the drugs, doctor.
The addition of dantrolene has caused a slight decrease in the amplitude and frequency once again for the contractions, doctor.
Right on. That works perfectly. Great job, Dave. We got the contractions back to normal. Take a sleep at the patient and discharge him.
Sounds good.